RHAM, a DNA amplification technique similar to qPCR in its high specificity and sensitivity, was first proposed by Dr. Songyang from PlusLife. It has since been applied in pathogen detection for both human and animal health, environmental monitoring, and food safety. Compared to qPCR, RHAM can be conducted at a constant temperature and utilizes a highly efficient BST DNA polymerase, resulting in rapid amplification within 35 minutes. RHAM requires 6 primers, including 2 loop primers, to facilitate product amplification. The double-stranded DNA contains the target sequence, with the regions upstream and downstream of the amplicon labeled to illustrate primer binding. The reaction starts with strand invasion, where the forward primer F2, which is complementary to the F2C region, initiates synthesis executed by PlusLife's self-developed BST DNA polymerase, and the F1C has a reverse complementary sequence to the F1 region. Next, the forward outer primer binds to the F3C region with the sole purpose of dissociating the gray-graded DNA, which is now single-stranded. What happened with the forward sequence will now also happen in the reverse sequence, as primer B2 hybridizes with the B2C part and initiates the synthesis of the complementary strand. The outer primer hybridizes to the DNA strand, resulting in the dissociation of the upper DNA sequence. This will be our first product. The reverse complementary sequence F1 then hybridizes with the F1C part to form a loop structure, and the same process happens with the backward sequence B1 and B1C. This forms a so-called dumbbell structure. From this point on, all primers combine to generate more product, and the loop primer enhances product amplification. At the conclusion of each pair of new sequences produced by the RHAM reaction, a fluorescence probe hybridizes with the amplified sequence. The self-developed RNase H2 from PlusLife functions as a restriction endonuclease on the specifically hydrolyzes fluorescence probe, releasing fluorescence signals from quenched conditions that can then be detected by utilizing the Mini-Docs Light Indicator mobile app or PC software. We can detect, sample, and analyze fluorescence signal accumulation over time. If no signal is detected within 35 minutes, we interpret the result as negative.